sporogenesis completed. Now, anyone hear me?
Sir, you completed up to development of pollen grain from a microspore.
Development of pollen grain from macrospore. Did I discuss the role of sporopollen in germ pore? I think discussed. In a microspore, you know that the outer wall is axine, inner wall is intine. Axine
Yes,
is okay. Then I discussed pollen allergy.
No, sir, it was not discussed.
Okay. I think I discussed up to the role of sporopollenin and what the role of germ pore present in the pollen grain and also the pollen development from microspore, right? Up to two cells.
Sir. Yes, sir. Up to 2 cell stage.
Thank you. Now you see what happens here. There is a plant called Parthenium. Which is also called carrot grass. And this plant is responsible for severe allergy. And another most important thing, it is not an indigenous plant to India. Actually, this plant was imported here to India as a contaminant. The Parthenium is a plant, small plant, the seeds of which came to India as contaminant at that time it was imported from the North American country, the American country. And so along with this wheat, this Parthenium was carried to India and now it is widespread in the India. And it is these pollens of Parthenium is responsible for severe allergy. Now, one thing I should tell you here, basically allergens and allergy. Allergens may vary. Pollen grain is one of the allergen. Dust may be an allergen. Whenever any allergen enters the body, suppose an allergen, for the first time, when the allergen enters the body for the first time, if immune system of that individual is found sensitive to these pollen grains, then the immune system of that individual will start producing IgE antibodies. (415066). Who is that person?
Sir, IgE key.
IgA immunoglobulin. Just a minute, please. Someone enters. Now tell me who entered with the number 415066. Who entered with the number 415066, please? Please tell your name. 415066. Anyone enters? Just enter with 415066 in the class. Anyone? Otherwise, I am to send you to the waiting room again. Please, what I am telling you, 415066, someone enters. Please tell your name. Otherwise, I am to send.
একটু আগে তো স্যার আমি ঢুকলাম দীপ্তদীপ
নাম বলো।
দীপ্তদীপ সেনগুপ্ত।
না, দীপ্তদীপ তো তোমার নাম দিয়ে ঢুকেছো, আরেকটা ঢুকেছে 415066 দিয়ে। ও, দীপ্তদীপ সেনগুপ্ত। দীপ্তদীপ তোমার নাম দেখলাম মনে হলো একবার।
হ্যাঁ স্যার আমি তো নাম দিয়ে ঢুকেছি।
কিন্তু একটা 415066 এটা কার? অন করা আছে দেখি মিউট করবে। দীপ্তদীপ নাম আছে, আমি এটাকে বার করে দিচ্ছি, যদি হয় পরে বলবে। আমি আবার বলছি 415066। Okay. Now what happens then? The individual will start producing IgE antibodies. What happens next? Will bind to mast cell. If it is a mast cell, IgE antibody bind to it. So no symptoms appear. For the first entry, for the first time when any allergen enters, the body for the first time, no symptom will appear. But when the same allergen,
স্যার কন্টামিনেন্টের পরে কি লেখা?
contaminant with imported wheat. When same allergen enter again, this allergen will bind to the IgE antibodies attached to the mast cell, so that permeability of the mast cell changes and histamines are released. Just a minute, please. Okay. কি ব্যাপার, কি নাম? নাম লিখে না তো, আপনি সেন্টারে বেশি আপনার সেন্টারে যাবেন, সেন্টারে রামনগর 3 নম্বরে, রামনগর 3 নম্বরে আপনার অক্ষর খোলা আছে। 3 নম্বরে গিয়ে আপনার নামটা বলুন, উড়িষ্যা দাস। ফার্স্ট ক্রসিং থেকে মেইন রোডে, ফার্স্ট ক্রসিং থেকে মেইন রোড দিয়ে গেলে বাহাতে 2 নম্বর বাড়ি, দুটো বাড়ি পড়ে, দেখা আছে ইনস্টিটিউট অফ কনসেপচুয়াল বায়োলজি। হ্যাঁ শুনেন, একটা ফটো নিয়ে দেন, ফটো নিচে আনছেন নেই, দেখাতে কুস্তি কোন পেজটা খালি আছে, এই তো দিও প্রতিকা বুঝিনি কথাটা, আমি ফোন করে দিয়েছি আপনাদের, আমরা জানো আমি কি এখানে ভর্তি হয়েছি নি? अच्छा उनको थोड़ा सा पूछना से केवल फोन करता हूँ आपने आई लो आपको अपना हम जानी है ये देखिए फोन पे क्या है ना ओके histamines are released from mast cell and allergy is caused.
স্যার হিস্টামিন স্পেলিংটা কিভাবে?
HISTAMINES. Okay? Complete it.
স্যার ডায়াগ্রামটার মধ্যে রেড ডটটা কিসের?
Oh, yeah, allergen. Allergen binds to IgE antibodies. Clear? Completed?
Yes, sir.
Okay, again I am telling you, another student enters 415066.
স্যার 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 নিলয় চক্রবর্তী স্যার
হ্যাঁ?
নিলয় চক্রবর্তী স্যার নিলয়
আচ্ছা বুঝতে তো নাম দিয়ে ঢুকছো না কেন তুমি?
স্যার এটা স্যার ডিস্টার্ব করছে স্যার নেটটা স্যার প্রচন্ড স্যার
আচ্ছা ঠিক আছে নামের জন্য ওয়েট করছিলাম নীলা চক্রবর্তী ঠিক আছে
হ্যাঁ স্যার নেটটা স্যার
Now, is it clear up to this?
Yes, sir.
Okay. Now you see that's why the Parthenium grass, the allergens released, actually pollen grains released from it can cause allergy in this way. Now we are rubbing it out, please. Is it completed?
Yes, sir.
Is it Orithro, do you hear me? Orithro,
Yes, sir.
you please do one thing. You take a photograph of the participants and send it to me, please. Can you?
Okay, sir.
Thank you. Covering all participants, please. Can I rub it out now?
Okay, sir.
Yes, sir.
Next part is viability of pollen grains. Viability means functional ability, efficiency. How long pollen grains is found viable? Generally, you know, depends on prevailing temperature and humidity. Viability of pollen grains depends on prevailing temperature and humidity. That means temperature and humidity, you need to remember this point because it's coming in the entrance. Prevailing temperature and humidity. And you need to remember the viability of some pollen grains. That was viability of pollen grains of wheat and rice. Pollen grains of wheat and rice. Viability 30 minutes. Pollen grains of members of Rosaceae, Leguminosae, for it is about 1 month. So viability of pollen grains vary plant to plant. Acha, one thing I'm telling you, just for 2 minutes I'm coming. Wait, and complete it, please. Okay. Now, so viability depends. Now you see pollen grains can be preserved. Pollen grains for a long time in liquid nitrogen at minus 196 degrees centigrade temperature. And this is called, this type of preservation is called cryo preservation. Pollen grains can be preserved for a long time in liquid nitrogen at minus 196 degrees centigrade. This type of preservation is called cryopreservation. And by cryopreservation, not only the pollen grains, different types of other living cells like sperms, egg, embryo, seeds can also be preserved for a long time. And whenever required, they may be taken out and they can be used in different purposes. So this is what about the pollen grains. Now, you know, pollen grains are rich in nutrients. You know, pollen grains are nutrients. Hence, they may be used as food supplement. Hence, food supplement. And it is coming in the entrance. Pollen grains are rich in nutrients. As they are rich in nutrients,
নিচের পার্টটা দেখা যাচ্ছে না
they will be used as food supplement. Clear now? So you need to remember this. That's why now pollen tablets may be available in the form of pollen grains may be available in the form of pollen tablets, pollen syrup. Sometimes some sports person are you found to take pollen tablets or syrups, as it is believed that it may increase the efficiency of the sports person. So this is what about the pollen grains. What I've learned today, how are pollen grains formed? Then I see that what are the different layers of the pollen grains, the role of each, how pollen grain can be fossilized, what is the role of germ pore, what about pollen allergy, then you know viability of pollen grains, two factors controlling it, then coming to the viability of different pollen grains, how can pollen grain be preserved for a long time, and what such preservation is called. And then obviously the pollen grains are rich in nutrients, so they may be used as food supplement, and this is coming in the entrance. And also pollen grains may be available in the form of pollen tablets, you know, pollen syrup. And sometimes some athletes are found to take pollen grains, so this is because it is believed that it may increase the efficiency of the sports person. So this is what about the pollen grains. Now we have to proceed towards the megasporogenesis. Now, can I wrap this, please?
Yes, sir.
Thank you. The next part is...
Sir? Sir, I am having a question.
Yes, tell me.
Sir, why Parthenium is called carrot grass?
Basically, if I am showing you the Parthenium, then you will see why it is carrot grass. Just a minute, please. Just a minute. Just a minute, I'm showing you. Look at this Parthenium plane. Is it visible to you?
Yes, sir.
Almost similar to the carrot. Have you seen the carrot?
Yes, sir. Yes, sir.
That's why it is very similar to it. That's why the term is used sometimes. Okay.
Okay, sir.
Now coming to the next, the pistil megasporogenesis. And I think you can remember this is the thalamus. Ovary, style, stigma, together pistil. So if you draw it in a larger way, and you know inside the ovary ovules are found. Number of ovules vary plant to plant. Now I think I discussed in class 11 the placentation, the mode of arrangement of ovules in the ovary. What about the ovules present inside the ovary? Now you see, generally pistils are of two types. One is called syncarpus, other is apocarpus. In case of syncarpus, you know, here the pistils are found to be fused together. Pistils are fused together, then you use the syncarpus. Pistils are free, then you can call the term apocarpus. Now you see, what about the ovules present inside the ovary? Look. Look at this diagram. Here, this is the nucellus. Inner integument, the outer integument, is the nucellus. Here, this point is called micropyle. So you can call it micropilar pole. And this is the Chalachal pole. And here inside which you will get the embryo sac. Now I want to show you how is embryo sac developed.
স্যার চালাচাল পোলের নিচেরটা কি কোনটা?
Or nucellus. Nucellus. Clear? Now I want to show you how is embryo sac formed. So can I rub this part? Can I rub this area?
Okay, sir.
আউটার ইন্টিগুমেন্ট তার নিচের ওটা কোনটা ওটা?
Yeah, outer integument is the outer covering. This is this one. This is the inner integument, right?
স্যার
Here-
নিচের পয়েন্টটা মাইক্রোপাইলা হচ্ছে।
Micropilar pole, micropyle. The aperture found here is called the micropyle. So now I am showing you how is megaspore formed. How is megaspore formed? Look, if I am going to show it, you see here. So nucellus, and this is called megaspore mother cell. Inside the nucellus, there is single megaspore mother cell. Is it clear? Next, what happens? Megaspore mother cell undergoes meiosis. After meiosis 1, you will get this called the dyad. Dyad megaspore. It is haploid. Then meiosis 2. Now you see out of now it is called your tetrad megaspore. Out of 4 megaspores produced, one is larger and found functional, and this is called functional megaspore in most cases. In most cases, functional megaspore is formed. A such type of development is called actually monosporic development. So single megaspore single functional megaspore will give rise to embryo sac. And this type of development is called monosporic development. When a single functional megaspore gives rise to embryo sac, this is the embryo sac. Then this is called the monosporic development. So here what happens, I am repeating it again. In the center, in the ovule, you will get the nucleus. In the center of the nucleus, only one megaspore mother cell is formed. In the center of the nucleus, only one megaspore mother cell is formed, which undergoes meiotic division to form diet megaspore followed by tetrad megaspore. In most cases, out of 4 megaspores produced at this stage, 1 megaspore is found larger and it is called functional megaspore. And this functional megaspore then undergoes development to form the embryo sac. And such a type of development where from single functional megaspore the embryo sac is formed is called monosporic development. And you see in this case, other 3 megaspores which are to some extent smaller in size will ultimately degenerate. In case of monosporic development, single functional megaspore will give rise to the embryo sac, and other 3 comparatively smaller megaspores will ultimately degenerate. Is it clear to you all? This is up to the megasporogenesis. Megasporogenesis is actually up to the formation of megaspore.
Sir, embryo sac কোন জায়গাটা?
Embryo sac means this megaspore is actually embryo sac. This megaspore transformed into embryo sac. It becomes larger to form embryo sac. Are you getting me? Or the functional megaspore grows to form embryo sac, right?
Yes, sir.
The embryo sac is also haploid. Now, can I drop the upper part?
Okay, sir.
Completed this part? Okay. I'm just rubbing this one. Now you see the fate of embryo sac. You see what happens in the embryo sac. This is the embryo sac. Undergoes free nuclear division. Your nucleus divides, but there is no cytokinesis. হ্যাঁ তো বন্ধা কোন কেন আমি কি কোন হ্যাঁ কাজ করতেন না আজকে আমি দিয়ে দিতে পারেন লোন আমি তো আমরা দিয়ে দিলাম দিয়ে দিতে পারেন লোন বহুত কি করো বালিটা বাজিনি আমি বালিটা ভাড়া থেকে অংশ দিচ্ছে ভাড়া কিছুটা দেয়নি কিছু টাকা আচ্ছা কিছুটা বলা কিন্তু যেটা হবে দিতে পারবেন বুঝলি তো বন্ধা কিছু টাকা ভাড়া এগুলো আছে না কোথাও দিয়ে দেওয়া হবে এটা দেন এগুলো কিছু টাকা হবে Okay. Further free nuclear division. You see, again, another free nuclear division, giving rise to after that,
স্যার
yes.
স্যার ফোর্থ স্টেপে নিউক্লিয়াসের সংখ্যা আরো বেশি হবে না?
Sorry. At one step is extra. Sorry. Okay. Now you see here what happens. There are two airs actually. Three cells are formed in the Chalaja land. This is the Chalaja land. Hello. আমি তো ক্লাসে আছি ইয়ের জন্য তো কোশ্চেন পেপার নেওয়ার জন্য নাকি 10টার দিকে আসুন 10টা বলছিলাম 10টার সময় জন্য হ্যাঁ Now this is the arrangement. You can call it the so here these three are called antipodal cells. These two cells are called, these two nuclei are called polar nuclei. This is the egg. And these two are synergids. Together called egg apparatus. So in this way you can call it, this is the central cell. These are 7-celled, 8-nucleate stage of embryo sac. In this way, 7-celled, 8-nucleate stage of embryo sac is formed. Is it clear now? You know, in the embryo, first of all, whenever the single nucleus present in the embryo sac undergoes free nuclear division 3 times to produce 8 nuclei. After the formation of 8 nuclei, 3 nuclei are shifted to the Chalajal pole and they are bounded by a membrane forming 3 cells, and these 3 cells are called antipodal cells. 3 cells are shifted to the micropylar pole. Among the middle larger one is called the egg, and the both side of the egg you will get the antipodal synergids, and egg and synergids together form the what? Egg apparatus, and 2 nuclei are found in the center called polar nuclei. In this way, 7-cell, 8-nucleate stage of embryo sac form. Is it clear?
স্যার সিনার্জিডস কোন জায়গাটা?
These 2 cells are these 2, this one and this one. Are you getting me? Okay, I'm showing you again. Suppose I'm rubbing this part. So if it is a larger embryo sac, then you see now you see here this one is egg and these 2 cells are the synergids. Is it clear now?
Yes, sir.
Again, here you will get the 3 cells, and they are the antipodal cells. In the center, you will get 2 nuclei, they are the polar nuclei. Clear? Is it clear now?
Yes.
How is 7-cell, 8-nucleate stage of embryo sac formed? The most important question for both board, and several questions even coming from here from the entrance also. These synergies may give rise to some thread-like structures or special thickenings called filiform apparatus. That means synergids would give rise to special cellular thickening at the micropylar end called filiform apparatus. The role of filiform is to guide the pollen tube toward the embryo sac. Filiform apparatus the role of this filiform is to guide the pollen tube towards embryo sac. And the synergy cells are the cells which are provide seating arrangement to the pollen tube entering into the embryo sac. The role of synergy is here to provide seating arrangement to the pollen tube entering into the embryo sac. After that, synergies will degenerate. Is it clear up to this? Megasporogenesis completed, and you have idea about the, just remember, egg apparatus is formed in the micropylar end, and antipodal cells are formed in the chelazal end. You need to remember this thing. Antipodal cells are formed in the chelazal end, and egg apparatus is formed in the micropylar end. Clear?
স্যার উপরের পার্টটা একটু দেখা যাচ্ছে না।
ঠিক আছে? Clear?
Yes, sir.
Okay. Can I rub it out?
Yes, sir.
Yes, sir.
Now next, so once the microsporogenesis and megasporogenesis is completed, the next course of action should be the gamete transfer. The next phase should be the gamete transfer, and for this the process is called, pollination. The first the transfer of and third to the stigma of different flowers of same plant, different flowers or same flower or from anther to the stigma of same flower or different flower of the same plant or different flower of different plant. Now you see, suppose Both plant A and B belong to same species, to different plants, but they belong to same species. So you know, this is called autogamy. This is called so in case of autogamy, pollen grains are shifted to the stigma of the same flower. So first of all, autogamy. Same flower. In this case, transfer of pollen grains occurs from anther to the stigma of same flower. Then you can call it autogamy. But to facilitate autogamy, two conditions are required to be satisfied. Remember this thing. To facilitate autogamy, two conditions are required to be satisfied. One, pollen release and stigma receptivity are required to be synchronized. Release and stigma receptivity are required to be synchronized. Point 2. Anther and stigma are required to be very close to each other. Anther and stigma are required to be very close to each other. So this is what about the two conditions which are required to be satisfied to facilitate autogamy. Normally in the flowers where the normal flowers where actually anthers and what it is called anthers and the stigma are well exposed. The type of flower where anther and stigma are well exposed, the chance of autogamy is very rare. The chance of autogamy is very rare in this case. Are you getting me?
Yes.
Because actually, you know, the why actually plants is not preferring autogamy, because if continuous autogamy may lead to inbreeding depression. As you know, same plant is involved in the production of seed continuously. So there is a chance of inbreeding depression, so that the efficiency, productivity of the plant may be decreased. So that's why plants are actually trying to avoid these. That's why in this chapter you will see there are some outbreeding devices which are developed in plants to avoid autogamy. So because the chance of variation is very less in case of autogamy. That's why the plants are trying to avoid it. But sometimes when pollinators are not found, then they're forced to undergo autogamy. That's why autogamy is there also. Now you see.
Sir?
Yes.
Sir, what is inbreeding depression?
Inbreeding depression means continued inbreeding leads to decrease in the efficiency of the plant, or efficiency of the plant, productivity of the plant. And this is called inbreeding depression. Are you getting me now?
Yes, sir.
Now what happens, you see, now there are certain plants. Like oxalis and Commelina. Two types of flowers are found. Two types of flowers are found. One, chasmogamous flower. In chasmophagan, anther and stigma are well exposed. Other is clistogamous flowers. These flowers will never open. These flowers do not open. So as clistogamous flowers do not open, so here anther and stigma are found very close to each other. So self-pollination is assured in case of clistogamous flowers. Self-pollination is assured because here anther and stigma are found very close to each other and flowers even not open. So this type of flowers, so you can say most important term when coming in the entrance or even in board. Are invariably autogamous. Clistogamous flowers are invariably autogamous. Why? Because here these flowers do not open and hence anther and stigma are found very close to each other. So here only option is self-pollination. Only option is self-pollination in case of clistogamous flowers because these flowers do not open. As these flowers do not open, there is no scope of cross-pollination. So only option is self-pollination, and hence it is said clistogamous flowers are invariably autogamous. And therefore these flowers advantage they produce assured seed set because self-pollination is confirmed. But for the disadvantage, there is a chance of inbreeding depression, because here continuously inbreeding is going on. As separation is going on continuously in case of Clistogamous flower, chance of inbreeding depression is there. Chance of inbreeding depression is there. Is it clear to you? Is it clear?
Yes, sir.
Okay. Now there are some plants showing both, as I said earlier. So you see, in a plant, sometimes you will get these are called Chasmogamous flowers. And these are called Clistogamous flowers. So in the same plant, whenever pollinator is available, then Chasmogamous flowers facilitate cross-pollination. And whenever pollinators are not available, then Clistogamous flowers come into act so that seed production can be assured. So this is the advantage of a plant having both Clistogamous and Chasmogamous flowers. Is it clear now? You know, in Chasmogamous flowers, both anthers and stigma are found to be exposed, but in Clistogamous flower, where it is found, the stigma. In case of Clistogamous flowers, what happens? In Clistogamous flowers, the stigma and anther are found very close to each other, but they are not exposed because such flowers, that is Clistogamous flowers, do not open. As Clistogamous flowers do not open, so there is actually scope of cross-pollination is not there, and hence, you know, there is continued self-breeding or inbreeding occurs, which that's why it is said. So one of the disadvantages is that it may lead to inbreeding depression. But the advantage is that, as you know, that the anthers and stigma are found very close to each other, hence self-pollination is assured. So these flowers produce assured seed sets. So this is the advantage. Is it clear to you?
Yes, sir.
Okay. Now coming to the J2-nogamy and J-nogamy. একটা দাও হ্যাঁ হ্যাঁ দিছি তোমরা আছে কত টাকা কোন কত দিলো বানাবো আলু কত Okay. Next comes to the J-tonogamy. In this case, transfer of pollen grains occurs from in this case, transfer of pollen grains occur from anther to the stigma of different flower of same plant. But the chance of variation is to some extent more, but also the same day. So you can call it is functionally and genetically genetically autogamy. Clear? So it's clear now why some question comes why J-tonogamy is considered functionally cross-pollination? Because in this case, two different flowers are involved. Because here two different flowers are involved, that's why it is genetically self-pollination. Functionally cross-pollination. It is genetically self-pollination because two same plants are involved here. Because two same plants, the same plant is involved, two flowers of the same plant involved, hence it is genetically autogamous. Are you getting me? J-tunogamy is functionally cross-pollination because two different flowers are involved in pollination, but it is genetically cross-pollination because
Self-pollination.
Self-pollination because here two flowers of the same plant is same plants are involved. Clear?
Yes, sir.
Okay. Now regarding, so here the chance of variation is 2 section more than that of the autogamy. Clear? Then comes to the last one, the genogamy. Occurs anther stigma of different flower of different plant. Okay? It's around this side. Different flowers of the different plant. So here the chance of variation is maximum. If you compare J-tunogamy, genogamy and autogamy, chance of variation is maximum in case of genogamy. Chance of variation is minimum in case of autogamy. Is it clear to you?
Okay, sir.
So types of pollination completed. Now next one is actually agents of pollination. Wind, water, and animals, insects, bat, birds, snail, etc. So you can classify these wind, water, animals in one group, and among animals you will get the insect, bat, birds, and snail. Now you see so clear. So in my next class I am to start from here. Okay, this is next Thursday. Okay. Okay. That's all for you. Thank you very much.
What we will do, online or offline?
Online, now online, until further order. Basically, as per government rule.
Okay, sir.
Okay, thank
Okay,
you.
sir.
Thank you very much.